Problems being here on Friday due to another course.
Yeah, I have one, so can it also be recorded on Friday, or I have to ask the other course to be recorded?
Which course is that?
It's from the other master program, so yeah, I know, I understand, but I really want to do it.
I think from applied machine learning, the last one is also double.
The last lecture?
The last
lecture.
Yeah, there's only one lecture.
So that will be, we can sort that out a bit later then at least. We'll have to see. You know, it's hard to deal with all of these things. I just try and organize, but then. So the course that is in the main competition here, we always give to the education office to say, look, you know, avoid a clash here. But this year that didn't happen. Didn't work, I should rather say. Okay, so anyway, here you see some almost all lovely people. They will be the teachers in your course. One of which you might recognize. That's a gentleman here in the front. That's Jose. He will help you to the best of his abilities in getting you through the assignments pertaining to this course. Anton will do one lecture for me when I'm in Paris. Anton, as you've met him probably, those of you who have done fundamentals in bioinformatics have seen this man. I hope you did at least. Otherwise something is wrong, this guy we don't talk about. This is Chico, who will also be, he's one of our PhD students, will also help out during the practicals. And here we have Bas, another case of PhD student, who is, will also help out. But you know, these two guys here do the bulk of the work. He will be there only a bit for the last assignment on hidden marker models, but come to talk about that in a moment. Here we have one guest teacher this year. We'll fill you in on copy number variation and all kinds of interesting things happening at the genome level. There will be an assignment attached to that, so you really run software that is also used in the clinic, for example. So to give you a feel of the importance of the sequence front. Okay. One little experiment. Who of you is not in the bioinformatics and system biology master? Are there any students here that are not in the master? Ah,
the only one.
Very good. Well, special welcome to you then. What's your another one? What's your background?
Computational science.
Computational science. Very good.
I did computer science. Now I'm an exchange student here.
Okay. But your background is also computational science. So you're sort of same as guys, right? In that sense, at least. Okay. Very good. Okay, here's the outlook of what you'll have to endure during the coming 2 months sequence analysis. A couple of lectures, as you can see, the right-hand column here, you see the practical work. So basically, Mondays, Friday afternoon, you have a lecture in this building on the Mondays, in the other building, the science building, on the Fridays, and the practicals will all be in the other building with one of the longest hallways of the whole of Europe. This is the champion building, right? It's fantastic. And let me help you there. Anyway, the lecture flow is as follows. So a little bit of an introduction, and as I'm speaking, we start with an alignment algorithm, dynamic programming. It's not totally unknown to you, I hope, at this stage. So I think we can do those steps quickly. I'll finish. There are 3 ways of alignment that I come to talk about: that global, semi-global, and local. We'll do all of those. Then we go into evolutionary models, what models are there to entertain, and we'll embark on similarity, exchange matrices, describing evolution. Then we go into BLAST, but a bit more than you've seen so far, a bit more on the algorithmics of BLAST. Then you get the special lecture this year for the first time on sequencing, on the reference genome sequencing, and reference-based sequencing, I should rather say. And another program that's all about complementary variation. We do a practical here, you can see that. Here you see the practicals. We'll start in the afternoon with a paper exercise on dynamic programming, just as a little reminder that you're going to carry out, you really make your own method, your own algorithm. You'll hopefully feel proud when you've done that, like I created something that can be used and could be used by you in the coming years. And so then we have the second practical won't be programming on your part. You'll carry out, you'll take more of an application view, a user view, but you'll answer some questions on that. And then the second assignment is the largest of the 3. is on hidden Markov models. We're going to do some prediction, training an algorithm on data, running it and see what happens. These are the 3 practical lines, and these lectures are there to support the work you have to do. So, as I said, we will do the blast type practicals, then this practical, then we go on to multiple alignment. Multiple alignment is defined as an alignment of more than 2 sequences, 3 or more, and there are all kinds of algorithms that you need to know about. Then we go on with the Markov models. It's an important general tool, a statistics-based tool to really formulate problems in a mathematically sound way and carry it out. And with all kinds of software, and you're going to put in snippets of code to make that happen. So creating, once again, your own with a Markov model and training it. Phylogeny, there is one of the main applications, of course, of sequence analysis is understanding evolution and using that then to maybe remediate something or do something in the cell based on that knowledge. And I'll finish off this year with a lecture on what's there to be entertained in comparative genomics. How to use this stuff, what kind of patterns do we see if we cross-compare all kinds of genomes. This is here on the slide, but there is immediately a disclaimer, of course. Let's say Canvas, the new glorious tool, this year for the first time in use at this university, will be leading. Yeah? So if there is a day change, so don't come to me and say, yeah, but on this slide it was said. No, Canvas is the thing to look into, in case of doubt. Okay, so a bit more on the practical assignments. So we'll start this afternoon with pairwise alignment, dynamic programming. We'll do reference genome alignment as the second practical, DNA copy number variance analysis, and then the third one, as I said, is on hidden Markov models. Because the third is the last one, the weight, the end, how do you get your mark for this course? There will be an exam, a written exam, counts for half your mark. The other half is for the 3 assignments, and here is a division. You see that the third one is a bit larger, so has a bit heavier weight than the other 2. And all of this should add up to 100 and number 5. Yeah? The main course book is a book by Richard Durbin and others. He's at the Zanger Center. Sean Eddy is one of the early adopters of hidden Markov models in biology. The Hammer software suite is made by him. So these guys are really in the know. And if you read the book, you'll see that these guys love hidden Markov models. It's written, it transpires that is in their mind. But it's the standard books in sequence analysis. So. And it's a relatively cheap book, and that's always also maybe a bit of an... although it can be made very cheap, I can tell you. PDF format, I say no more. Okay, anyway, I'm not allowed to tell you this. Then there is the book, some of you, or many of you, I hope, might already have, the book that was also recommended for the earlier course, Understanding Mathematics, the big one, and a lot is there to be found about algorithms in sequence analysis. Now, we should start talking about the Human Genome Project, of course, and so on, but I won't do this so much in this course. I'll just say that all the hope we have that will prove that we are absolutely different and take an extreme position, certainly when it comes to intelligence and being social compared to other species such as animals. I have to sort of sober you up a bit. And people thought, we have so many letters in our genome, it's roughly 3.3 or 3.2 billion, that must truly be the largest. Well, here we go. This is where we are. We're just a mediocre lot. A water lily is 30 times bigger when it comes to the genome. A salamander laughs at us. Okay, we're bigger than a bacterium. Oh, that's great. And then a virus will win. That's the smallest, right? And the virus, it's even debated, is that a life form? It really needs a host to do the thing. So it's not generally regarded as life. Then you talk about the smallest are single cell organisms such as bacteria. Here is genome size. So here, sorry, I can't make it any better. I don't have another slide, but then people said, okay, size, but the number of genes, surely, that's the real use, then we should win. No, also there, we're just mediocre. So there is nothing special about us in the genomic sense. Okay, we have to live with this, be good, and this is still good, without even being champions at the genome front. Okay, another thing that was a big discovery in 1976, You won't remember, you remember, you don't remember. You were not there, most of you. No. And so there was a splicing, the idea that DNA was being discovered, and this is a string of letters, and people had an inkling that would carry some information, perhaps, the cell was doing stuff. And then it was found out or discovered that when it comes to taking a section, some of the information of DNA, I had no clue until that time how it really worked. How is that converted in information to build a protein? One of the words there, and I'll get to that in a moment, is translation. This is a nucleotide sequence. You know by now probably that is 4 letters roughly. And proteins have 20 letters, and the translation needs to take place. I think I go fast. The two computational science people, if I go too fast, do something. Yeah? I'll talk about translation as if you know it. If you don't, give me some signal. Okay. So here you see then how this really works. Some kind of pasting. You see that some parts of the brown bits here are sort of glued together in the mature messenger RNA molecule, the intermediate molecule that carries the information to build a protein to translate into a protein. And so that means that some pieces are used and others are not. And there is a very sort of intricate mechanism, machinery to all do this. The main idea is all of the engines that do all of these things, take that together is all proteins basically. And proteins are the doers in the cell. And how do proteins come about? Another graphical image of that. Simple one. DNA makes RNA makes protein. That was the main line catchphrase from 1976 onward. Here you see this conversion from a nucleotide sequence, 4 different letters, 4 nucleotide types, via groups of 3 called codons, is translated into one of the 20 letters. This is a famous double helix discovered by Watson and Crick while they were drinking lots of beer in some pub in Cambridge. So people take immediately the strategy to get good in science to go to that pub and drink themselves silly. Sometimes it even works, but not for everyone. So in here we have a protein sequence as a result of that mechanism. Because we have to go from triplets, and how many possible triplets are there if you have 4 letters? Who can do quick mathematics here? Very good, 4 to the power 3. So here are the 64 possibilities. And you know all of this, of course. What is apparent if you look into the, so this is the nucleotide, the codon, the 3 group, and this is the resulting amino acid. There are 20 types. And on a quick scan you can already see something interesting, that many codons, here is one, there is one, that in fact the 3rd position in the codon in the group of 3 does not matter at all. You just get the same amino acid. Meaning, because you have 64 places to code for 20, well, you should take 3 off. Why is that? Who knows? 61 stop codons, right? They are separate beasts. Well, here is one. So this is not translated in any amino acid into sort of a stop sign. You have 3 of those. And then the start, who sees the start? Where's the word start? But you know, right?
The
Met is the start. So with what letter necessarily does every protein start? Met. Do we see that? No. And why is that then? The first bit of a protein typically is being cleaved off. That's sort of an address tag. And in the cell, that is very often taken away. Okay, so you're in the know there. That's very good. And that means selection pressure on codons. When it comes to coding for proteins, you'll see that the selection pressure is less on the 3rd codon position as compared to the first one, right? And you can find that back, and gene prediction, all of these technologies. I won't use this course, this is on really comparing, make use of that fact. Okay, so basically what we do is the main thing, and you've probably heard that message to some extent already in the fundamentals of bioinformatics and principles of bioinformatics course. But a lot of what we do is exploiting signals and patterns that have arisen during evolution. Yeah? And the most important thing is that we think there is something called divergent evolution. So a lot of species and a lot of molecular components find their origin in some common ancestor, and from there on went to diverge. And that's why the phrase divergent evolution, from a common ancestor, once upon a time we were all the same, we start sort of moving away from one another. And the problem there is that some species or some components move away faster from one another than others, and that's of course depending on selective pressure. And Darwin was the early, the discoverer of all this, and so it's certainly these patterns, they vary wildly among molecular families and so on. And that makes it a little bit difficult sometimes to use these signals well. Okay, we're just a few minutes in the lecture, but here comes already some stern remark. You have to know this by heart. The chance is 98% that you get a question on reproducing these 4 terms on the written test. And even, I tell you this, 98%, roughly everybody, always, chance is near 100. Half of you will know it. I still don't understand it, but I say it once again. There's a near certain probability that this question will be asked. Give me the thing. So even if you know these terms, it's also sort of comes in handy when you know a bit what it means. So I'll talk you through. And this holds for living organisms, this holds for computer programs, this holds for optimization techniques, which work very well based upon this evolutionary principle. So basically you need something that evolves. We call that a template. The beast, the gene, whatever it might be. Then we need to move along some axes, and in evolution that's much time axes, and we talk very often about generations, children, grandchildren, and so on, at all kinds of levels. So the idea is that a template molecule, such as DNA, will be copied into next generations, and all of the research has proved to us that this really happens. Then something interesting should happen. Life is totally dependent upon error. Without error, there is no life. And error leads to something important, that is variation. So, I don't know if everybody here is in the know of the little game, children game, kiddies game, Chinese whispers. You know how that works? I give this man a sentence on a piece of paper. He whispers it, reading it in your ear, and you whisper what you hear in the next ear, and that goes on. And then at the end of the classroom there, we ask the lady in the corner, could you say what you heard? And we all compare how much similarity there is with this first line that was spoken, whispered here, and you'll find out very little. This is what you get in copying errors, right? And so that is also what happens in evolution, although the error rate is surprisingly low in living systems. 1 in a billion letters might be copied incorrectly. That's stunning. That's very, very good. Anyway, if you do this very many times, and over long periods, you'll see a gradual but unavoidable change, which can lead to the diversity we observe on this planet. Right? I already mentioned the name. Of course, Darwin was the man who really thought this and put this in a framework to think about. He was against his times because religious feelings were taken to be against all of this, and so he found that it's important. Your fitness, how can you survive? And from that, a lot of evolutionary theory has evolved. That's come of that. And selection, of course, is the important word. So, with those 4 ingredients, you will have evolution. It can be over millions of years, billions of years. We have 4.2 billion years. That is the approximation of how long evolution takes place on this planet. After the first life form abounded, came about. But it also works at the bacterial level. If you do an experiment and you wait 2 hours, you already can observe evolution. Bugs have changed. They became more resistant to salt or less. After a while, they can even start giving some color, lighting up. If you do the proper gene technology, you can do all kinds of things over a number of generations based upon these evolutionary mechanisms. Okay, so be in the know. So template, copying mechanism, mutation, leading to variation, and selection you should know by heart. Okay, so basically evolution and divergent evolution, Darwinian evolution starts with a common ancestor. Associate from now on as common ancestor in this course, and then you will two genes might happen. This might be in a cow, and this might be a gene in a horse, and you get to divide. They go live in different environments. One environment is warm, one another environment might be cold, you get adaptation to those environments, and that means changes will arise within genes that are related to that sort of functionality. So, homology is the common term for this, and homology doesn't mean anything else than the fact that there is a common ancestor between two genes or species or some entities you compare. And it's an all or nothing. You are homologous or you're not. Okay, so then if you compare, and here you see a schema of two genomes, so the genome of one species as opposed to the genome of another species. And now, because you have common ancestry, you could talk here about the gene that was the same thing in the common ancestor. Yeah? And they might become different, but there are, of course, genes in two genomes between orthologous and homologous species that refer directly to the common ancestor. We call that an orthologous relationship. It's a biological definition. Those two genes that are the same thing in the common ancestor. Now, in the genome level, a lot of dynamics happen, and I'll talk a bit more in detail about that in a moment, where you can have all kinds of copying and paste activity. What you see very often happening is that a gene is copied somewhere else in another position in that very same genome. It could be on the same chromosome, it could also be on another chromosome, and things like that. You call about if you see that. And there is, of course, a homologous relationship, meaning if this is a direct copy of this, then obviously this refers back to that common origin. You will see that you get variation more easily if we do that. But that is called a parallelous relationship. This is another thing I'm afraid to say you have to know. Know the term homology, know what it means, know orthology, orthologous in between species, and know parallelology within a single genome. Right? It's not so difficult, but it's really vital to understand. If you read any book, you don't know this. You'll be in trouble before you know. So here once again. Now you can start doing some fun, and this is an early textbook example about globin genes and some pattern you find. This could be a bioinformatics tool that you run, and you've got a so-called phylogenetic tree. You've seen those pictures a lot ever since Darwin. And here you see then an ancestral globin gene. And then you see that you get two forms, alpha and beta. So we're talking about alpha and beta, say here. And then you see this is happening, these two genes happen in three different species. A frog, a chick, and a mouse. And if this is your pattern, and you don't know this type of stuff, how would you reason about it? So the reasoning is you have an alpha and a beta chain. So what could have happened? You have a gene, an ancestral gene, and then in one species it could start. You could start very early on. Have this situation, and then you get two things happen. I should tell you. You have a copy, of course. So there is a copying event like this, alpha to beta, or beta to alpha. We don't know, but it doesn't matter. And you have speciation. You got three species here, and everything had a common origin. So you get speciation events and something we call duplication events. So from alpha to beta is a copy event, a duplication event, and you have the speciation event. So this here and here is called the speciation event. And now you need to reason what happened first. What's the most likely scenario that happened? So you find three species and they all have they all have an alpha copy and a beta copy. Then you got speciation. What would have happened if you would have found a copy, the beta copy in the mouse and the chick, but not in the frog? Then you could, or if you find only in the mouse and not in the chick or the frog, what would your reasoning then be? And then you would probably say, first speciation, and then in the mouse you got a duplication, right? So, depending on what you see, your data, the model you build will depend on that. And that's I guess, an important point. And this happens all the time in evolution, these type of things. So this is very, very common. Orthology and parallelology, both are flavors of homology. Okay? And another one is, this is a bit scary sometimes in the bachelor course. I say, you stand in the elevator, it happened to you here, and you stand even for a long, certainly in front of the elevator, you stand for a long time, but also in the elevator. And then you come out and you've already changed, because somehow the genes of your laboring elevator mate, so to speak, came through the side or some effect into you. That's what happened with bacteria. Maybe we're happy that this doesn't happen with us. But okay, that is basically, so it's not sexual, it's asexual, and it's really, but this is the main reason of the most scary thing happening on this planet, the risk of microbes becoming resistant to medication, right? Because they exchange genes very, very rapidly. So very quickly, if some bug gets resistant, other bugs in your body will become resistant to it. There's a high likelihood of that. That's really a problem. And early on, and this happens often in science, you know, there was the idea like, yeah, this seems to happen, but then, you know, it doesn't fit nicely in what we think we understand and our models, and then you just deny, you just say, oh, this is not real. You know, you deny the reality of it. But then, of course, it's now so abundant that people, there is so much abundant proof that this, so we have to deal with it. You have to say, yes, it happens. And by the way, it's highly dangerous if, when it comes to resistance of bacteria. Yeah? So this is horizontal transfer, also called lateral transfer, as opposed to vertical transfer. So through the generations, like how we do it with children, grandchildren, and so on, is called vertical transfer. Yeah? And of course, in species, there is a combination of this. And also in us, you might say, you know, we have many bacteria that are sort of, you know, really us almost. Yeah? They're in our gut, and it all happens. So in us, it also happens. Okay, so that's important as well. A bit more on parology. This is sort of a Darwinian experiment. Think along with me for a moment. You have selection pressure support. You suppose you talk about a very important gene. You die if it doesn't work properly. So that means there is high selection pressure. You should not change that gene so that it doesn't function anymore, because then you will certainly not create any offspring anymore, no children, and therefore no grandchildren, so the gene will be lost. So the idea is that evolution hardly happens. There is a lot of selection against evolution. Now at a certain point in time, you get this copy from alpha into beta. So at that moment, you have two, say the copy is complete, you have two workable copies of that same gene. What is happening now? And then you get an evolutionary play garden in a sense, because what happens if the beta gene starts changing? Is that a big problem now? The answer is no, because alpha is still there to do the job. Okay, so you know, selection pressure goes down, no problem. But you know, here you see this moment that say gene beta starts changing, starts moving gradually away, maybe from the function it was doing, and then you see all, you know, if the function becomes different, there is at the end, of course, the possibility that the beta gene is doing something completely different, right? No lecture to the second half, first lecture, I should say. So here you see, you know, some examples of some databases. I'm going to gloss over, but you know, most of you will have seen some of those at least. I guess Uniprot might have been opened by you, and so on. So have a look if you want. I go on showing you a few things. This is an alignment. You might have seen this. I don't know what's a bit self-righteous, but anyway, my family name has amino acids. That's easy. And here is the DNA alignment. Yeah, so this is pairwise alignment. Two sequences just being lined up, and then you check, and here an asterisk is placed under positions that have remained the same, so that have been conserved over the course of evolution of these two sequences. Okay, so it's all about comparing sequences. Analyzing has a lot to do with comparing. Ever since Darwin, we have the comparative analysis. To just take various life forms and learn from comparing. So you can, of course, to compare things, you can use many properties. When you talk about a protein or a DNA sequence, you can say, okay, how many, what's the fraction of alanine in the protein and of guanine in the DNA. You can measure things like isoelectric points. You don't need to understand it. Physical thing, you can measure a number per protein sequence, per protein, so you can compare based on that. Or you also, you know, have the molecular weight. You can use this, or all kinds of morphological characters. You know, how many alpha helices are there in the protein, and so on. You can do all kinds of things. But if you really want to learn fundamentally, and you know, and sequence alignment means you need to use the molecular evolution. And you get a window on that using the sequence information. And as those of you who have done the last course, Fundamentals of Bioinformatics, have already worked on this principle. You could call it a lazy investigation. Somebody brings in an unknown gene or an unknown protein sequence, asks you, can you find out about it? And you say, yes, sure, no problem, I'll run BLAST for you, right? A program that's finding stuff out about the DNA or a protein sequence. And the idea is that you use homology. If, BLAST finds a sequence in the database with high similarity, you might start arguing, oh, it's so similar, my guess is these are even homologous. And then something interesting clicks into place, because then it's likely that all kinds of features that one entity has is shared by the other entity, by the other protein sequence. And then if one of those two is an unknown sequence, you've learned about it. And that's the basic idea. What you see here. And it's all under the constraint, and this is just a warning, I guess. Yeah, so homology is a binary property, right? It's yes or no. You cannot be a little bit homologous. It doesn't happen. You're homologous or not. You have a common ancestor or you don't have a common ancestor. Okay. And here is that reasoning. If you have an unknown gene G, the moment you run BLAST or anything that suggests to you that gene X and gene G might have a common ancestor, the trick to say all I know about G is shared by X works surprisingly well. Yeah? Because 98 or more % of the cases this works correctly. This works. So that makes it nice to do sequence analysis. You really can use it to find out about molecular properties by inference. That's very important. This green arrow that we can do by inference used on based upon available data. Okay, so if you want to reconstruct, you do this. You look at sequences, you look at identities and the similarity of sequences. If you think there is homology, then you can start finding out about function, and you have to always keep in the back of your mind that how does function come about? There is a sequence that encodes a structure, protein sequence encodes protein structure, encodes a protein function. Also at the DNA level, of course, you have that the DNA sequence encodes the DNA structure, and therefore the DNA function, but it's a bit less clear how to get a handle on that, because most of DNA has a very regular shape and a double helix, so there less sort of tractable things happen for us, and this is why we use this mostly at the protein level. Okay? Here a little bit of illustration of that fact, evolution, and things that are important to a protein. Here you might see a yellow bit, see that, and then we reverse the protein. You look now, and now we do this. And then in this case of the protein, isocitrate dehydrogenase. Should have been A there, apologies. This is the green, the yellow bits are the active site. That is where really the protein binds something, is doing its thing. And the color coding is that, they have compared a number of protein sequences that fall into this structure. They've looked at the conservation by means of, is there conservation in the alignment? And the color coded is on this structure. And I hope you'll agree with me that there is, around the yellow, there is more blue than red. If you agree there, then I can make my point. That you see that conservation, so what is functionally important, stuff around the exact active site is more conserved than bits further away, like here and there and here and there. Yeah. So, Darwin's point, what is important tends to be conserved. Yeah? Don't change a winning team, is the American way of rephrasing this, basically. Okay, so mutations and what might happen with this. So I'll talk you quickly through all kinds of mutations. Oh, I don't know why this is so animated, but anyway. So a point mutation, already mentioned that's a single letter change, right? So for example, if this is your original sequence, I think it's Scottish. We is small and Scottish. And so here, do you spot the single change somewhere? You see that, right? Okay, good. So, second base. Who has an idea what this is? Everybody here? It's a mutation that leads to frame shift, but then you have to know what a frame shift is. That is computational biology. Do you know frame shift?
Yeah, when you put an extra gene,
Roughly.
so the following part of the sequence is shifted.
It's a codon that is shift. But I can fill you in really at the end of this lecture if you want my details on it. So frame shift mutations is just a mutation that changed the frame that would indeed lead to a totally different protein sequence in practice. So here you have one, and then the shift would be. So what do you see? What happened here? And this is what leads to a frame shift. You see here the register 3, 3, 3. And because here you see that something happened, what is happening? There is an A is doubled up here. And that means that your frame is now, the A frame is gone and the B frame is all. And this leads to a fundamentally different protein, right? Frame shift mutation. So frame shift mutation is something that elongates or shortens a protein by a non-3 multiple. Yeah, so 1 comes in, or 1 falls out, 2 come in, 2 fall out, 4 come in, 4 fall out, 5 come in, 5 fall out, 7 come in, so not 3, 6, 9, because then you just would put in or take out a complete codon. And that of course does something to your protein, but your frame stays intact, right? So frame shift mutation changes the frame. Okay. Now, and so frame shift mutation can be a result of this, of a deletion. And deletions can be short, a single letter, or very long, complete domain. And here you see a little example of that. Okay? And if there is a deletion, there is probably the opposite. There you come. Where you see that a bit here came in between. All of this happens any split second in your body. Yeah? So what more is there to do? So this is mostly interesting at the DNA side when you get an inversion mutation. Some of you might see it, or it's hard to stand in front of the blackboard. So some part of the sequence is red probably. Not fat. So here is an inversion. B, 8, cat. Okay, so the whole thing is there. See that? Look at it at home. It takes just an hour, then you'll find the inversion. Yeah, yeah, that's what it is. Okay, then there is another one. These are mutations that might change the protein sequence, but what they also do is they change things such that the expression, the way the cell produces the protein, for example, how much of it is affected. And that can also be very important and devastating. And these are so-called DNA expression mutations. But these ones, we will not really focus on it because we don't see it so much at the sequence level. And we are talking here about sequences. Yeah? So the main problem is, we all talk about common origins and very important common ancestry, and that's so important, but normally we don't have the stuff anymore. The common ancestor is lost 2 billion years ago. It's not there. It would be great if you would have this sequence with the descending sequences from that common ancestor. But we don't have the common ancestor. And here is just a simple example of a common ancestral sequence, ABCD, that changed in one species into ACCD, so B changed into C here, B into C there, and here you see that the sequence ABD, because the letter C fell out, the deletion of the letter C in the common ancestor led to ABD here. Now, what we normally are confronted with is the problem that we have these two sequences, but not the ancestor. So we need to start guessing what happened in evolution by comparing sequences. And you might agree with me, if you have these two sequences, you compare them, you align them, this is alignment. You might say, they start with the letter A and they end with the letter D. You know what, it's likely that that is conserved. So I put the letter A, so there was a letter A in the ancestor, and there is a letter D in the ancestor. And now you have to guess what happens with these two, this region, CC versus B in the other sequence. Then there are two possibilities, in this case, short sequence, either this or that. And then you need to say, when you have this aligned, like, oh, which one do I prefer? And maybe you would agree with me, if you have to score this with one minus sign and here a minus sign in another position, but it's just one minus sign added to make it in register, that's basically what you do. This is not an amino acid, it's just something we put in as a placeholder to compare the sequences. It will be difficult to tell, you know, which one is the preferred one, which one is the correct one, biologically, by the way. The left one or the right one? The left one. B went into C. Right? That's this one. And it was the first C. Okay? So, this is so basically that's a problem. We need to compare sequences. We want to use common ancestry, but we don't have the common ancestor. There is a guess part in all of our reasoning. And to stress that point a bit more, here's another example. This is then, you know, of a certain position in an alignment. This is DNA probably. There was a G as a common ancestor, and one sequence kept the G here, and another sequence changed to a C. We only will see this, and we will say the reasoning is, I see it's not conserved, so one mutation took place, which would be correct. You could even, you know, with 50% chance, you might say this might be the common ancestor, this is the mutation, then it would be wrong. And you know, you have all these type of possibilities. This could also have happened. You see, you think minimal reasoning is, I see an A and a C, probably one mutation. You would be wrong here because both letters appear to be mutated. That's possible too. And here, you will say here, I see here an A and an A. You know, my model is nothing happened. It's just a conserved A. And in reality, this is biology. There was a G originally, and they both changed into an A. Can happen. And here's another one. This is a favorite by many biologists, the so-called back mutation. When you have, you know, this one was conserved, the G remained the G, but here it went to an A and to a C and a T and then into a G. And you would think, hey, I see no change, I see a G here and a G there. That's just a conservation. This is a lazy position, nothing happened. Very active during evolution via back mutation. Yeah. Okay, so that's the sort of the tricky component, because we typically will not have the common ancestral sequence. Okay. Now let's still talk about divergent evolution. This is just, you know, a little picture you might put in your mind to think about this as a framework. A common ancestor, one sequence and another sequence. They start changing, they start diverging. Here it's cold, and this is this environment of this sequence, of this organism in which that sequence is warm, and you get adaptation and differences. So these sequences become dissimilar. Then you have this important thing that sequence encodes structure, encodes function. Very often in evolution you find that although the sequence might become different, very often you see that a structure doesn't change so much as a result. Basically, even at the functional level, if you have a protein, you can change 50% of the amino acids of a protein, ballpark figure, nothing will happen. The protein will just do what it should do. The problem is you need to know what 50%, of course, right? So you can easily ruin the protein, but if you do it well, there are enough places that don't matter so much. And of course, knowledge means you need to find out where that is, how this positioning is. So, sequence structure, and there's a famous sentence by Tom Blundell, structure more conserved than sequence. If you look at this with a mathematical eye, it's not so well defined, because you have a sequence space with letters and you have a structural space with molecules in 3D coordinates. How can you say this? How do you compare this? That doesn't compare. So, but anyway, basically this is the story. Sequences change all the time, and very often the structures remain more similar. As a result of structural conservation, very often you also find that function is less affected, so not all mutations will immediately lead to a functional change. Yeah? There is conservation at that level. And that's important to realize. So function is normally pretty much preserved within orthologous families. How do you think this will, when we allow paralogous sequences in our sequence set? What do you think? Does it get better defined, or are we getting more tricky? Do you remember what I said about the paralogs, the play gardens? Selection pressure goes down, the second copy can go off on its own change. So then you bring yourself a bit more into trouble, because paralogous sequences could well have changed their function more easily. So the best is, if you really want to fingerprint, is to use orthologous sequences. And then of course is, you have to find out what the orthologous sequences are. Yeah? Okay. This is the word of warning on divergent evolution and homology. It's a Boolean thing, because even in Nature papers I've been reading, like, you know, this family is more homologous than that family. Nonsense, right? There is no more. You are or you're not. Okay, so far I've talked about Darwinian evolution, divergent evolution, things coming off a common ancestor. There is also another class of evolution. People think it happens a bit less often, and that is that, you know, almost, if this is divergent evolution, conversion is a bit like this. You know, you have two different molecules and somehow they start assuming the same function. And they still remain very different. Very often that happens, and I think that's somewhere here, that a small sequence, a stretch of sequence, that are responsible for the function in one protein is sort of transplanted into the other protein, and somehow that protein therefore is going to carry out that exact same function. So that is so. There are all kinds of examples, and a famous textbook example to give you a bit of a feel at the sequence level is this one. This is these are proteinases. These are a bit tricky proteins. These proteins, they chop up all the proteins. So you have to be very careful when you have them on board. They're in our stomach, but they are with pre-enzymes. They only are made active when they're really needed, and then they're shut down again. That's very important. So there are different families, and so what you see is there are 3 amino acids mainly responsible for the function, the cleaving function, and this is called the catalytic triad, the 3 amino acids. And you see in the chymotrypsin it's there, it works, it chops up proteins. It also works in serine proteinases, but you see that even the order of the 3 amino acids is different. The same type, but different order differently, and here another permutation of those same 3 amino acids. It is as if the spacing is the same. That is not true either. So how do you find this out? By comparing sequences. How will you find out that, oh, this is very important, you know, there is no conservation or nothing, you're cooked. Also, these important letters are just very few letters in long sequences. So to find this out is really hell. It's next to impossible. And there is one other fundamental point I'll get to in a moment. Here is some home reading for you if you want to find out more. Here again is the picture. So there is no common ancestry. So, you know, I don't know if this makes any sense to you, but before we had this common ancestor, this is just to show you that they don't originate from the same thing. They're just different. So the sequences are way different. When you have convergent evolution, very different sequences. The structures remain way different. If they started off as being different structures, they keep on being different. But then by transplanting or copying or whatever happens, a smaller bit of sequence, or these 3 letters, for example, as in the example, that makes that one structure will start doing the same thing as the other structure or the other sequence, if you like. Yeah? So that is convergent evolution. It happens less often, and there was one thing that can happen. A moment to go back to the slide, and then you may ask your question. Yeah?
But is it also possible only by selection, pressure? For instance, flying with different organisms.
Yes, okay, of course, it can be selection. It can be selected, that's true. You know, if there is a requirement of flying, this would really help if you get it. That's why it can then become conserved. So after these proteins are there can be selection to keep it, absolutely. And from that point on, you could talk about divergent evolution again. So all of this, of course, relates to one another. But is it a fundamental function? And one of the things people, you know, want to give you an idea of this, it's also referred to as non-orthologous displacement. What can happen? You have a species A and a species B. There are genes in there that are conserved divergently. They have the same function. And now suppose that in one of those species there is another protein, totally different shape, that assumes the same function as that divergently evolved function. And that means then that now you have 2 different components in the cell that's doing the same thing. Then maybe if then the divergently... Okay. We can do that, it makes it clearer, I guess. So you have this common ancestor, A, B. So here is a sequence and it's doing the same function, F and F. And now here there is another beast, comes through the back door, another protein, C. And somehow C, by convergent evolution, is assuming the same function F. And now in this species, this one species here, the species says, oh, then maybe this sequence gets lost. It's not necessary anymore, it disappears. And that means then basically this one is replaced. This is the orthologous gene of A, B is orthologous to A, and now you see that it's superseded. C has taken over the function and B is lost. And that is referred to as non-orthologous displacement. The ortholog is replaced by a non-orthogonal, by a convergently evolved protein. Okay, then this. Okay, yeah, and also think, so if we do sequence analysis, sequence alignment, we have a common origin in mind, a common ancestor. And that means alignment is basically only defined for divergently related molecular components of sequences. Yeah? So you need a common ancestor. And and I mentioned already analogous sequences. They can become very distant because they do that easily, and then they become a challenge for proper. Yeah? So basically, don't align analogous sequences. It doesn't mean so much. You can find a motif, that's another thing. Oh, is there a motif? Because then you can find out about it. But relating it back to a common ancestor is not relevant. Okay? So, what have you got? I showed you this weak-cat example. So there are substitutions, insertions, deletions, duplications, inversions. Basically, the algorithms to compare sequences only do the first two blocks. We only use these. If we say this happens perhaps less often, duplication happens a lot, but inversion at the protein level doesn't happen very often. So how can we align? What kind of alignments are there? And here you see this. This is a mutation. This is maybe a deletion of the letter C in this sequence. And if this is a deletion of the letter C, then these two letters are an addition, are an insertion of GNA. Right? Insertion is the opposite of deletion. You need to know the common ancestor to find out what is what. Was this letter, did that letter fall? Still we need to start scoring alignments. And so that means if you score an alignment, here are two alignments. Both are not so good, I think, but anyway. Scoring should prefer reasonable or biological alignments, if you like. And what they have to, if you score, these things can happen. There are three possibilities: an insertion, and a deletion, and a mutation or a match here. This is conservation, but it's comparing two, this is probably DNA, so two nucleotides. So we need to score matching with matches and mismatches. This is a match, and we need to score something that we refer to as gaps. Minus signs are referred to as gaps. So we need to be able to score. Now, I'll show you a slide on this in a moment, and what are the scoring schemes to score matches in either DNA or protein sequences. Basically, I'm already giving you a forecast of gap penalties. A gap penalty comes with a length. So here you see two gaps of too long. Two minus signs is a gap of size of length 2, and here you see a number of gaps that all are 1 in length. The gap here is 1, gap of 1, and so on. So gaps can a gap can be 100 long, or 200 long. In DNA it can be thousands long, if a whole chromosome drops out, for example. So the gap size varies wildly. Now, we do more in a moment, but this is sort of the scoring. We do some simple type scoring based upon the length of a gap. You see that we have sort of simple functions, and basically in our algorithms we only will use this one or this one. And these biologists always want curved lines. They don't like straight lines. In biology, everything is always squiggly and so on. So, and that is then referred to as a concave penalty, where what you gain, the penalty, the points off you gain are really dependent upon the length. The advantage of these curves is you don't need to know how long you are because it's linear, right? For this otherwise you need to know. Okay, so basically then. And we'll get more into this in more detail. This is easy to implement, this is a little bit harder to implement, and here there is no really efficient solution. The algorithm will become a lot slower if you want to do this. But more about this when we really start implementing and talking about the algorithm in a moment. Okay, so now this is about gap penalties. When we compare the letters, matches or mismatches, you can do this with DNA sequences. And most algorithms just use this simple scheme. They say if a letter is the same, you get a point. And if a letter is not the same, you get a point off or 0 as a point. Both happen. Just, similar, the identity.